Polypeptide synthesis model by Louis J. This is the key for all the components within my model.
A polypeptide is a long linear chain composed of amino acids linked together by peptide bonds. The polypeptide chains fold and combine to form proteins, which are simply a functional chain of multiple polypeptides, crucial for structure, function, growth, restoration, and coordination of the body's tissues and organs. Thus, the process of polypeptide synthesis is essential, involving the formation of polypeptides. This process occurs in 3 stages: transcription, translation, and protein synthesis. Polypeptide synthesis begins within the nucleus of the cell, in which DNA is contained. DNA is a type of nucleic acid made up of a long sequence of nucleotide pairs linked together by hydrogen bonds to form an elongated double-stranded molecule. These nucleotides are each made of one phosphate group and a sugar, known as deoxyribose, which form the backbone, as well as a nitrogenous base of either adenine, cytosine, guanine, or thymine. Transcription is the first step in polypeptide synthesis and must occur as the original DNA, which contains the genetic code for proteins, is too large to exit the nuclear pore, necessitating the production of a smaller complementary single-stranded nucleic acid, mRNA. To start, initiation involves the enzyme helicase attaching to the promoter, a specific sequence of DNA located at the origin of the DNA molecule, unwinding the double-stranded DNA helix into 2 individual strands, a template and non-template strand, as helicase has the structural capabilities to do so. Then, during elongation, the enzyme RNA polymerase moves downstream, with the 2 strands reforming naturally behind it, to restore the original DNA double helix, reading the template strand 1 nucleotide at a time, building the mRNA molecule out of 3 complementary nucleotides which pair with the chosen DNA strands. Cytosine and guanine nucleotides pair together, while adenine and thymine nucleotides also pair. However, anytime RNA places a complement to adenine on the DNA, it will place uracil instead of thymine. Following this, termination occurs when the RNA polymerase transcribes a sequence of DNA nucleotides known as the terminator sequence, causing transcription to cease in order to prevent unnecessary DNA information in the mRNA. Just to note, at this stage, the mRNA strand is known as pre-mRNA. Some further complex processing steps such as splicing are required to eventually produce the mature mRNA strand, which can then leave the cell's nucleus through the nuclear pore and enter the cytoplasm, where it will attach to a ribosome. Thus, the mRNA is ready for the next step, translation. In the cytoplasm, there are many transfer RNA or tRNA molecules floating around that each carry an amino acid and 3 complementary nucleotide bases known as an anticodon, which are attracted to the ribosome after transcription. To begin, during initiation, the ribosome reads sets of 3 nucleotide bases on the mRNA molecule, known as codons. These codons are each associated with a respective amino acid, with the type of positioning of these amino acids determining the end product, being the protein. The process of translation can only be initiated when the start codon, AUG, is read by the ribosome, as seen in the codon chart. After reading the start codon, the ribosome binds the complementary start anticodon, UAC, to it, thereby depositing an amino acid, in this case, methionine. The ribosome will then read the next codon and bind the respective anticodon of the next tRNA molecule to it. Once paired, the tRNA will deposit its amino acid, whereby a peptide bond is formed to hold the amino acids in sequence. Then, the first tRNA molecule and its anticodon detach, leaving behind the amino acid. After, elongation will occur, whereby the ribosome moves along the mRNA molecule, allowing further tRNA molecules to continue this process, lengthening the amino acid chain. Finally, termination occurs when the ribosome reads the stop codon, UAA, UAG, or UGA, causing the chain to detach, and thus leaving the product of a polypeptide. This polypeptide then develops into a protein, which is a functional chain of 1 or more polypeptides, after undergoing some extra processing steps to become functional, such as folding on itself. Polypeptide synthesis can produce primary, secondary, tertiary, and quaternary protein structures, reflecting the increasing complexity of proteins, from simple amino acid sequences to fully functional molecular assemblies. Types of essential proteins include enzymes, such as helicase, which act as biological catalysts, and antibodies, which defend against disease, encapsulating the significance of polypeptide synthesis.